In order to run the analysis in this paper, open our Quantitative Fast FLIM FRET analysis package found in our supplemental information. So data is organized as shown in this supplemental figure, where each replicate is divided into subfolders within the experiment. We're going to copy the replicate analysis folder into the folder for each replicate. I'm going to do the analysis for one replicate in the BH3 swap screen as an example. The replicate analysis is divided into several steps. The first step is to run the compression code to compress the raw INO TIFF into a smaller size. This is an important step for long-term storage. So we're going to run this compress INO GUI version 2 code. And the graphic user interface will pop up where we will select our input folder. This is the folder where we will find our raw INO TIFF, which we have organized here. So I'm going to copy and paste the directory for the input, and I'm going to select an output folder. That means where I will save my compressed files. So I'm just going to create a new folder, select it, and click compress. And you can see the size of the files for each INO TIFF is saved as a multi-page TIFF. And the size is quite large. As the compression code continues, you will see files in your compressed folder, and you can see they're dramatically reduced in size. This script runs a lossless compression algorithm, so you don't have to worry about losing any quality of the data for future analysis. When the compression is complete, it will display the status on the GUI interface, and this may take 1 to 2 days for a larger data set for an entire screen. So I prefer to run this analysis while I'm taking the next replicate on a separate workstation. So once the compression is complete, I usually go and I double check that the number of files in my raw files equals the number of files in my compressed files. We have a notepad text file that pops up if there was a problem with compressing any corrupt file, and that will show as failed file names.txt. We don't have any failed file names here, but if you do, open the file, find the original failed file name, and copy it directly into the compressed file without compression. So that concludes the first step in the analysis. The second step in the analysis for each replicate is to run the protein gradient analysis, which extracts the relationship between intensity and concentration on the date of your replicate, using the standard protein images which we acquired on that date. So the first thing I do is move the INO compressed TIFF of the Cerulean 3 and Venus fluorophores into a separate folder. Then we open the INO client software, which can be found here. The INO analysis app asks you to load a data set. So from the standard protein gradient, I will open just any image. Then once the analysis view is open, we want to create an example of the analysis script. Then we're going to save the configuration for the analysis and run it in batch mode on the total data set. So I'm going to select the Emily NP fixed lifetime fit algorithm. I'm going to change 80 to 50. 6 tau 1 to 4,000. And most importantly, we're going to select the naive quadrant binning. So this style of RY selection just separates the image into 4 squares. Now the reason we did this is in order to ensure that we have no aggregates in our protein standard. Here you see none, but the analysis will extract the lifetime, the counts for each square, and then we will divide by the area of that square and get the intensity per pixel. If there is an aggregate that appears in one of these squares, then we will obviously identify that as an outlier in our standard curves. So that's why we do 4 squares in this RY selection. So the next step is to load your IRF for that date. So I'm selecting it for the date that I acquired. I'm going to load the dark. And finally, I'm going to save the configuration for the analysis. I'm going to save the configuration in the same folder where I have the standard gradient TIFF. And I'm going to save it as quadrant analysis. Then exit the INO acquisition app. So here's where we saved our quadrant analysis config file. And depending on the workstation, sometimes there is no local save directory designated, such as seen here. If that's the case, you need to add 2 quotation marks in order to avoid a future bug in running the next step. So once that's confirmed, we can open the INO FLIM hyperspectral batch analysis, where we designate the configuration for the analysis and the folder which we're going to analyze. So that's containing all of the files for the protein gradient. Then we select where we're going to save the results, and it asks where it should save a copy of the analysis config. I'm just going to save over the quadrant analysis we just created. And I'm going to save the results in the same folder where I've created the standard gradient TIFF file directory. So as the batch processing is taking place, you can see that a new folder appeared where our results are saved with the date of the analysis. And inside the results are being exported as CSV and TIFF files for each image processed.
Let's examine the data generated by the INO FHS software. We'll see that for each field of view, we get 2 files, a CSV file and a TIFF file. The TIFF file contains information about the segmentation map for that field of view. The CSV contains numerical information about each region of interest or ROI, And if we open the file for the quadrant analysis, we'll see that there is 4 ROIs. Each has its own unique ID. And any numerical information associated with that ROI is saved along the columns. So we'll see that the time resolution information is saved here. This is the TCSPC time bin resolution. In this case, it's 64 picosecond. The parameter estimates from the maximum likelihood estimation, the number of iterations that the algorithm require to converge to a solution. And if it successfully converges, then you get this OK comment here. The photon counts per ROI, the size of the ROI in pixels is also saved. Then we start to get into actually the data associated with the TCSPC channel. So here we get a TCSPC histogram, and I'm going to plot a few of these data. So you can see that this is actually represents a fluorescence lifetime decay. Again, and if we go down these further, we'll see the estimated lifetime profile from the fitting. This is convoluted with the instrument response function, or the IRF. With the IRF profile is also plotted here. A lot of this data you'll see is redundant, so there is a more efficient way to do this, and you can generate your own data. You can write your own scripts to generate similar information. Then once we start to get into information about the hyperspectral information, the hyperspectral channel, we start with the start wavelength. We start with 441 nanometer, then we go all the way to 851 nanometer. Then we start to get the spectral profile for the ROIs, and I'm just going to plot this so you can see what that looks like. And you can see here there's just a sharp peak in this region, and this, if we were to map the emission profile for fluorescein, we'll see that this will match it exactly. So again, these are the data generated for each ROI, and this can be generated without having the INO FHS software.
So the next step in the analysis is to run a script which combines all of these CSV files into a single Excel file, so it will be easy to plot the intensity versus concentration relationship. So we have created a MATLAB code in the replicate analysis folder here, which can be used to do this. So the first step, when you open this loop through purified protein CSV file, is to get the directory and paste it in this location, highlighted here. So our directory is the location of our CSV results. I'm going to copy this and paste it here. Now I will run the code, and when it's completed, a file will be exported, combined gradient results, which contains all of the results for these data here in one file. So here's the important result. This corresponds to the well label, so the well ID. This is the counts in the TCSPC channel, and these are the pixel-wise counts in the hyperspectral channel for the protein gradients. So the next step would be to label your wells with the concentration. I'm going to do that. Okay, so from my plate map, now I have imported the sample IDs. And for my the corresponding well, and you can see that I have added titles as well as the concentration of each fluorophore. So the next step is to plot the relationship between concentrations of Venus in the hyperspectral channel, counts in the hyperspectral channel, and concentration, as shown here. So you can see this is a nice linear relationship. We set the intercept to 0 and display the equation on the chart. Then we do the same thing for cerulean 3, so except for we use the count at time 0 in the TCSPC channel. You can see here the relationship again. Add a trend line, display the equation on the chart. And these are the slopes of the Venus and cerulean 3 gradients, which we will be using next in the following analysis. And voila, here is the final data. I'm going to leave this in the analysis folder as an example for your reference when you're doing this analysis. So the next step in the replicate analysis pipeline is to run the region of interest selection on our cell data. Our cell data, if you recall, is located in the INO-TIFF compressed folder, and we run the ROI selection using the INO FLIM hyperspectral analysis software. The first step, as you have done before with the protein gradient analysis, is to give it an example. As you can see here on the left, we have our TCSPC FLIM image, and on the right, we have our hyperspectral image of 2 proteins of interest here. And we're going to select our fitting parameters, though we don't use this fitting approach later on. It can be useful. Then we use an automated ROI selection. We select the NIHAD watershed segmentation algorithm. And you can see here that it identifies regions within the cell, and some regions outside in the background may be identified, but they will be removed in post-processing due to low counts. Now, any location in the image, you can click on an ROI, for example, and see the fit of that data. So the next step is to load the instrument response function, which we acquired on the same day. And finally, we're going to save our analysis configuration, as we did before with the protein gradient. I'm going to save it in the Select ROIs folder. I'm going to call it the name of the segmentation algorithm we use. So exiting the INO FLIM hyperspectral analysis software, our next step is to, again, ensure that the local saving cache was correctly inputted in the config file that we just saved. So if you open the file in Notepad, Once again, you see here my local save directory should be set to quotation. On some workstations, this will be automatically inputted, but for some reason on my computer, that is one thing that occurs. Run the INO batch analysis on the total data from the screen. We're going to load the configuration file, which we just created. Okay. I'm going to go to my replicate analysis, select ROIs folder. We're going to load the location of our INO TIFF. So here. We're also going to load a destination for where the analysis and the results will be saved. So I'm going to save that in my selected ROI folder. I'm sorry, I'm going to go back. I made one mistake. I'm going to click, when I'm saving the config, I want to click on the segmentation name so that it will be properly named when it saves the configuration. Then I'm going to process the batch. Here, with just those 3 example files, really it's not going to take long, but as I mentioned before, this can take a longer time for large data sets, so give it run this overnight and it should be completed for your screen. Now once this is finished, you will see the results are exported in your selected ROIs folder. And as you saw before with the protein gradients, each has a file that has been exported. But in this case, we have a lot more ROIs. Since you saw in the algorithm we chose, in the image, there were many ROIs within the cells. So that concludes the ROI analysis. So by the magic of video, I have run the batch analysis for a large screening data set in our BH3 swap screen rep 1. You can see I have a total of over 2,000 files here. And for each well, I have multiple fields of view. The purpose of the next step in the replicate analysis is to combine the data for each well. The instructions can be found in this notepad, and I'm just going to walk you through it. So the first step is to sort the data by type, and we're going to move all the CSV files into a separate folder. Then we need to open command prompt. So you can just type CMD in the Windows and hit enter, and command prompt will open. It chooses the C directory, but if your files are located in another directory, for example here, my files are in the E drive, you need to type E : and enter, and you will change the directory location. The second step is to change the path directory. So you're going to type cd space, then copy the directory to your CSV files, and paste in command prompt by right-clicking, and hit enter. To confirm that you're in the correct directory, you can type dir, enter, and you will see all the resulting CSV files in your folder. The next step is to copy this line of code from the notepad we've provided and paste it as before by right-clicking and hit enter. So the command prompt will go through each of the wells and combine all the CSV files into one file per well analyzed. It will continue until it completes all of the files for your entire plate. So this step might take 5 to 10 minutes to complete in command prompt. Once it's finished, you will see all of your combined files in the same location as your CSVs. The last step is to cut your combined data and move to a folder. This will be used in the subsequent analysis steps. The next step in the replicate analysis is to run the filter data to extract the bound fraction for each RY that was extracted from our RY analysis, and to generate raw binding curves for each well in our plate map. And speaking of the plate map, we need to create a master plate map as shown here. And I'm going to include an example in the example analysis folder for you, because the format of this is very important. So first, in the plate map, I have an example here. So for this screen, we have arranged our drugs by row, and we have designated our transfections. We have our cell lines designated here. We have 2 cell lines in this plate, and we have the location of our protein gradients, etc. But this format of our plate map is not compatible with the script. So we create a well-wise plate map as shown here. Every well, we show the row, column, the cells, which in our case are all going to be BNK / double knockout, or in other words, D3 cells. Our transfection, drug, the concentration of drug if applicable, and any extra information if you want to include it. So here, it's important to note that everything is labeled properly and that only the wells with cells in it are included in this well-wise plate map. So don't include the wells with your IRF or your Venus or cerulean 3 protein gradients or any empty wells. Just exclude them from creating this plate map. So that's pretty much the only rules, except for you need to indicate. So for our untransfected controls, you can see that for each drug in the plate map, we have untransfected wells and treated wells that are untransfected that will be used as controls to see if these drugs affect the lifetime or the intensity in the images, which could affect your results. So it's important to note that the word untransfected is spelled as this. with a capital U. And we also need to designate our treatment for DMSO wells. They need to be clearly labeled DMSO. Those are our controls for the positive and negative controls will be treated with DMSO only. What else? We need to make sure that our labeling of each drug is consistent. Don't have, for example, ABT 199 without a dash or with a dash. They always have to be consistently labeled in order to be analyzed as that group. Okay, so that's pretty much all the rules for the master plate map. We have the option to put replicate 1, 2, 3, 4 in case there's any differences made in preparing that plate. We give the option to do that, but if they're all the same as replicate 1, which often happens, you can use just one sheet for that, the analysis in your master plate map. Okay. Now that we have combined all the ROIs for each field of view per well in a single file, we need to filter the resulting ROIs in order to remove noisy data. And that's done in step 5 in this MATLAB code, which we're going to run the filter data exportation constant TX code. There are several areas here where the user needs to input parameters. The first is the directory to the plate map. And as you can see here, I'm going to give an example where I input my directory, including the name of my plate map, exactly as it appears here. The Excel sheet is labeled Rep1. That corresponds to what was indicated in the sheet name for the replicate that we're analyzing. We next indicate the cells that we have seeded in this plate. And in this case, we've seeded 2 BMK double knockout cell lines. So I need to directly copy the exact name that appears in the plate map as they are seen in the plate map, to ensure that the code knows what to look for in the plate. The next step is to indicate the path to your CSV files. Those will be found in the replicate analysis combined CSV folder. And ensure that you leave this last dash backslash when pasting your path. The final step to this analysis is to input your slope of the line for your Venus and cerulean 3 gradients. So we go back to our analysis we did earlier, and I'm going to directly copy the slope for our Venus and cerulean 3 protein. Finally, I'm going to save the script and run it. This may take an hour or so to run through the total data. And what this script will do is export the filtered raw curves in a folder within folder 5. So it will happen. So the first thing you'll see that's exported in the raw curve is what is called the untransfected phasor coordinates. And this is used later. It is the reference phasor coordinates for the untransfected position in the phasor plot for each treated untransfected well. This file will be used later to determine whether any of the drugs affect the donor lifetime, which is an important consideration for the analysis. So as this script is running, you can see the filtered raw files are exported in your raw curves folder. Now, this is a warning. Do not open these as the MATLAB code is running, because if you do, they may save over top of each other. Once the analysis is complete, you will see a file that is exported for each well analyzed in our screen plate. I'm going to open one of these files where we know the two proteins bind. For each region of interest listed in the rows, we have several features which we've exported. Total concentrations are listed in A and B, whereas free Venus in that ROI is calculated in C. By plotting columns C and D, we can generate our binding curves as shown in the paper. You can see at this stage we've plotted the entire range of concentration of free Venus in every ROI. In subsequent steps, we will be filtering all the data such that it's less than 50, so this data will be removed because we have no confidence in our estimation beyond the point that we sampled in our original protein gradient. So I'm going to just cut out that data. In this, you can also see we've calculated the FRET efficiency from the phasor plot analysis, and on the right, we've exported features from the INO FLIM hyperspectral software, which is showing you the bound fraction calculated from the double exponential fit, and you can examine the effect of using different fitting algorithms to calculate bound fraction if desired. So you can see what the binding curves may look like by analyzing the data in a different method. However, we found the fitting algorithm with the phasor approach to be more reliable, so all subsequent analysis will be done using these data sets. If you would like to compare that data set to a negative control for collision, so this protein has a mutation in its binding site that prevents binding. You can see that the increase in collisions occurs in a more linear fashion compared to the positive control. So at this point, we've confirmed that we are observing binding for positive control. The last thing you can do with this at this point is decide on your filter for M-cerulin 3 concentration. So we've exported the total data from 0.5 to 8 micromolar cerulin 3 expressed in each ROI, but we require the designation of a narrow range before doing binning in the next stage. Our default is to filter between 1 and 3 micromolar, but depending on your cell line, you may have different range of expression. So in step 5, we have also exported an untransfected phasor coordinates file in the raw curves folder. If you open this, you will see the plate map copied here for only our untransfected wells. The purpose of this is to see whether the drug treatment at each concentration, whether it affects the coordinates compared to our DMSO controls. However, in order to see this in terms of a lifetime value, we need to convert the cosine and sine coordinates in the Fourier space to a lifetime value. And for this purpose, we will run the next code. So you can find that in step 6. Open the get lifetime from phasor coordinates file in MATLAB. This script is very easy to run. The only thing you need to import is the path to your folder with the raw curves as shown here. I'm going to copy and paste the path within these brackets and run. When the analysis is complete, it will show complete in the command window, and we can exit the script. You will see an exported file in the location 6, and for each well, we now have the calculated lifetimes for the treatments listed. The next step is to bin our raw curve data exported in step 5 by free acceptor concentration. The reason we do this step is to equalize the contribution of each replicate when we combine the data in the next steps for analyzing the combined replicate. So open the unshifted raw curve binning. Here we have a few steps to take. We need to input the path to our plate map. So I'm going to do that here. The corresponding sheet for the replicate that we're analyzing is indicated next. For example, if you're analyzing rep 2 and you need to change this, you need to also update this. But for this example, we're doing replicate 1. Then you input your path to the raw CSV file where we exported the raw binding curve in step 5. I'm going to copy and paste this again. And importantly, I'm leaving the last dash when I'm doing this step. At this point, you can run the code with the default parameters if desired. And it will run and export the data in these 3 folders. So we will be using the phasor bin curves in the following analysis. But we also export bin curves with the INO fitting, that is the double exponential fitting, if it is desired to use an alternative fitting algorithm. And we also export the acceptor-donor ratio versus FRET for these bin curves, in case any user would like to look at that data in a bin format as well. It exports a donor concentration table, which tells you for each well in your plate map the average concentration of M-serline-3 in micromolar per well. This will be used later on to examine the variation in the expression across the plate. If you recall, in step 5, our exported filtered raw data looks like this. So we have the option of exporting a variety of binding curves depending on the type of analysis we did. So here in CD, we did a phasor analysis, and the bin corresponding curves are exported in this folder. On the other hand, we could have used the INO fitting. So if we wanted to examine the INO fitting algorithm, we would have plotted F and G, and this is exported here in the INO bin curve. Finally, we also export an acceptor donor to ratio versus FRET curve, and this is a traditional FLIM FRET analysis, which we do not use here, but may be desired for certain users. So within each folder, you'll find bins data with the well label. Let's take a look at the data for our positive control that we saw earlier. Here you can see each bin corresponding to the bins indicated here in our supplemental figure. So the raw data is binned by each free venous concentration, where we take the average of all the points within that bin. Here we showed the median bin point with the standard deviation at each ROI. And we did the same thing with this script, where this corresponds to the free acceptor or free venous concentration and our bin bound fraction at that concentration. We also show the standard error in the data, the number of points per bin, the free acceptor standard deviation, and the standard deviation in the bound fraction. If a row shows up as NaN like this, that means that at that point there were insufficient ROIs within that bin in order to calculate an average. We have a minimum threshold for, we require 5 points to consider the data good enough to analyze. So you can see the resulting binding curve for the bin's data as shown. The next stage of the analysis will require that we repeat steps 1 to 7 in the replicate analysis for reps 1, 2, and 3. Then we will combine the bin data for the matching, transfection, and treatments for all replicates.